So right now we're going to take a look at the last portion of the chapter 14 that we're going to be covering, which is the second line of defense. And what we talked about earlier was the ability of the cells in the immune system to be able to fight the infection once the infection gets past the first line of defense. And we mentioned phagocytosis as one of the main strategies that the body has and employs to eradicate infection. So phagocytosis is the process in which a phagosome engulfs a microorganism, for example, and eliminates it through digestion. So here, for example, we have a cell, which is a phagosome, and these are polystyrene beads who have been covered. And what is happening over here is the membrane of the phagosome is extending right over the target that is going to use. So at the end, as you can appreciate here in the second image, the cell has completely engulfed the target particle, in this case the bead. So the cells, granulocytes, macrophages, eosinophils, basophils, they're going to be survey the tissue compartments, and once they discover those microbes, those particle matters and those injured dead cells, they're going to ingest them and eliminate that material. And that is the process of phagocytosis. So oftentimes this leads to the recognition of immunologic information, which we're going to call them from now on antigens, as a foreign manner. Now, eosinophils and neutrophils, as we discussed earlier, are going to be very important in the eradication of bacteria by phagocytosis. So as I mentioned to you, the neutrophils are going to react early in the inflammatory response against bacteria, foreign tissues, and damaged tissues, and they are one of the main targets, main effectors, of the immune system to destroy bacterial infections. So oftentimes when a bacterial infection is happening, a patient will have a high neutrophil count in their blood. Eosinophils are another one of the granulocytes that are able to attack cells. They often are involved in the responses of the immune system against parasitic infections. Eosinophils play a minor role in phagocytic activity, especially when it's antigen-antibody mediated reactions. Monocytes and macrophages are some of the most effective phagosomes that we have. And as we mentioned earlier, those monocytes are going to transform into macrophages once they get out of the bloodstream and now invade a tissue. They also differentiate by increasing enzymes and increasing their capacity to destroy microorganisms by having an enhanced lysosome and other organelles needed for destruction. For example, histiocytes are a specialized macrophage that are going to migrate out of the blood, and some of the ones that are going to be included. We mentioned alveolar macrophages in the lung, we've mentioned Kupffer cells in the liver, dendritic cells on the skin, and macrophages in all the immune tissues of the spleen, lymph nodes, bone marrow, kidney, bone, and brain, the microglia of the brain in more particular. The following slide is going to look at the process of phagocytic recognition, engulfment, and killing. What I want to start you with is a short video about how a neutrophil, for example, is able to chase a microorganism, in this case Staph aureus, and it's able to then kill it. So what I'm going to show you here, I'm going to play the video for you, and we can take a moment to see it.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
So as we saw in the video, the neutrophil is able to sense the bacteria, and once it senses the bacteria, is able to engulf them. So this process is described here in the slide. So chemotaxis, which is the process by which the neutrophil chases the bacteria, allows the neutrophil to be close to the microorganisms that it wants to destroy. The microorganisms has PAMs, which are receptors on its surface that are repeated. And the neutrophil has pattern recognition receptors to bind to the PAM, and that leads to the adhesion of the bacteria. Now that the bacteria have been bound by the adhesion receptors on the neutrophil begins the process of phagocytosis, and you create what is called a phagocytic vacuole. The phagocytic vacuole now is able to completely close up, enclosing the bacteria around a membrane. A lysosome, which as you remember from the beginning of a class, it is an organelle that has a lot of the enzymes like lysozyme, DNAs, RNAs, and proteases and peroxidases that is able then to kill bacteria. The lysosome is able to fuse with the phagosome, which creates the phagolysosome formation, and eventually the enzymes present inside the enzymes released, by the lysosomes, and the changes in pH and the generation of oxygen reactive species inside the phagolysosome allow the destruction and the killing of the bacteria, which later the waste products are released outside of the cell as residue. So the PAMs, the pathogen-associated molecular patterns, are the molecules on the surface of bacteria that are able to be recognized by the phagosomes. They're able to generate signals that allow for the phagosome to be activated. And the cool thing about them is that they're only found, for example, in bacteria. They're not found in us mammals. An example of a PAM is the molecule peptidoglycan, which, are you familiar, it's the molecule that composes the cell wall. The lipopolysaccharide, which is also on the outer membrane of gram-negative bacteria, is another example of a PAM. And double-stranded RNA that is found in viruses also is an example of another PAM. So the PAM could be soluble molecules like double-stranded RNA found in viruses. They can be on the surface of dead microorganisms like the lipopolysaccharide or peptidoglycan. Of course, they are in the surface of living microorganisms, and many pathogens have PAMs that can then be used and allow the recognition by the immune system. So the immune system has the pattern recognition receptors, or PRRs, that are able to recognize the PAMs. And they're found on the surface of the phagosomes, the dendritic cells, and other cells like endothelial cells that facilitate the recognition of those foreign molecules and the foreign organisms that carry them by the immune system. So they can bind the PAMs, recognize them, and trigger the cell into action. One of the most important pattern recognition receptors are the toll-like receptors, which is a type of PRR. And they recognize a large variety of PAMs in many organisms, including bacteria and viruses, and they can set up the kind of signal cascade events that are going to activate the macrophage to allow them then to phagocytose the microorganism or secrete cytokines and other inflammatory modulators that are going to cause an inflammatory response. Now, the lysosomes, as we described earlier in class, are the organelles that are going to contain a lot of the enzymes that are able to destroy the microorganisms. So they then can fuse with the phagosome that is created once the organisms are phagocytosed and generate the phagolysosome. And therefore, you're secreting now all the enzymes and components found in the lysosome that are able to destroy the bacteria, often as short as 30 minutes following ingestion. So now we're going to look at two of the systems designed for destruction and elimination. And some of them are going to be the oxygen-dependent systems that are going to create myeloperoxidases to form halogen ions like the OCl that I have over here, or they're going to be the reactive oxygen species, the superoxide ion, the hydroxyl free radical, and the singlet oxygens, which we already discussed in class when we're looking at the lab, the superoxide dismutase enzyme, as well as catalase. The inflammatory response is one of the main responses following the recognition of a microbial agent by the immune system. And there are 4 different signs that are classical to recognize an inflammatory response. The first one is going to be rubor, which is the redness caused by the increased circulation and viral dilation that is happening in the tissue. That is the redness that happens in the area. Calor is the next response. It's the warmth that is happening in the area of inflammation resulting from the increased flow of blood. The tumor is the swelling of the area when fluid is now escaping the tissue from the circulation and bathing the area where the inflammation is to allow then the fluid to eventually go into the lymph and take the molecules that are causing the inflammatory response to be transported to the lymph node. And the last one is dolor, which is the pain caused by the stimulation of the nerve endings in the area. So again, rubor, that is the redness, tumor, that is the swelling, dolor, that is the pain, and calor, that is the heat. Now, oftentimes there's another 5th inflammation response, and that is the loss of function, the incapacity to use the tissue that is being responsive of the inflammatory response. All the signs of the inflammatory response are warning signs to the immune system that something has come in and caused an infection, and it allows the swelling. It's being caused by the extra fluid and the vasodilation of the vessels, which allow now the immune cells to come and infiltrate the area of inflammation to clear the area of microorganisms that may be causing the injury. So what is the inflammation going to be doing? Number 1, as I mentioned, it's going to mobilize and attract the immune cells to the site of injury. So in the picture that we have here on the right, you see a wound that is caused by a needle. The needle is bringing bacteria in. It's breaking a little bit of the capillaries, which now are vasoconstrict, and now that mediate immune molecules by the neutrophils and mast cells that are around that area, and that attracts cells of the immune system. Now, this set of motion, the next set of events that are going to cause all the 4 steps of the inflammatory response. When vasodilation happens, the vessels becomes bigger. That allows for leakage of fluid, which is going to cause the tumor. The excess flow is going to cause the calor, and the rubor is going to be caused now by the redness that is happening over there. As you can see, now you have infiltration of new neutrophils and other microorganisms, of other immune cells that are coming to the area, and they're helping then repair the reaction. So that allows the immune system to destroy the microbes and block the area for further invasion by producing a clot, and that clot eventually turns into a scab. Now that the scab is sealing up, we're going to have the edema apos formation. That is going to increase the area. The pus, it's a lot of immune cells that are now coming around, clearing the area, and some of them are dying as their way to destroy the microbes. Eventually, they begin to win the battle. Macrophages can then come in and help to repair the damaged vessels that are being that has caused part of the inflammation, and then you have the last part of the resolution and scar formation in the tissue. So there in green, what you have is the new healed tissue that is being formed around the area of the scar. So now, what we have then is going to be the steps of early inflammation, which is going to induce the areoles, the capillaries, and the venules, which are going to change to allow for the great release of fluid in the area. And that is going to be mediated by chemical mediators or cytokines. Cytokines are the hormones of the immune system that are going to be secreted by the localized immune cells that are in the area of the wound. So the vascular tissue is going to be able to secrete some of those chemical mediators. Now, some of them are vasoactive. They're going to affect the endothelial cells and the small muscle cells of the blood vessels to allow them to vasodilate. The chemokines or chemical factors will then attract the red blood cells that are in the blood to come and invade the area where the inflammation is happening, and those inflammatory mediators are going to allow to cause the fever, stimulate the lymphocytes to take action and destroy, and oftentimes prevent viral spread and they may also be the cause of allergic symptoms if the immune response is now out of control. So this table here, 14.1, shows some of the most important immune mediators and other cytokines that are released by some of the cells. What I would like you to do here is to just take a moment and learn them by groups. Look about the mediators of inflammation and immunity, the most important ones being interferons, interleukin-1, and the tumor necrosis factors. Other ones are going to be bioactive mediators that are going to change the vascular permeability of those vessels: histamine, serotonin, and bradykinin. And last but not least, you have cytokines that are going to regulate lymphocyte growth and activation. The most important of those are going to be interleukin-2, produced by B cells and T cells, and macrophage colony-stimulating factor. Interleukin-2 is produced by T cells, but it affects T cells and B cells. That's what I wanted to forget to mention to you guys. So again, take a look at the molecules in this table and see what are the general qualities that they have according to the three different types of immune mediators that are discussed. Now, the edema is the swelling that is going to happen, and that is happening because of the vasoactive substances are making the endothelial cells of the capillary to contract and form gaps, and those form gaps allow the leakage of liquid from the venules into the area. So you have the blood-borne components that are now going to escape into the extracellular space. Those are going to trigger an immune response, and the edema is that localized swelling and firmness because of the accumulation of that exudate in the tissue. So that is what's going to cause the swelling and the tumor in that area. Diapedicity is a mechanism by which the cells that are of the immune system are able to get out of the blood vessels and then migrate into the area of inflammation attracted by the chemotactic factors. So we're looking at the movement of white blood cells from the bloodstream into the tissue. And it is occurring because of the vascular endothelial having now gaps when the tissue is vasodilated. Of course, we need chemotactic factors. Those are the molecules secreted by the area of inflammation to attract the immune cells that are going to migrate out of the vessel, and by the process of diapedesis, snake through like an amoeba outside of the blood vessel into the tissue. As I showed you earlier in the lecture, chemotaxis, it's a mechanism by which an immune cell is able to follow a chemical trail that is going to lead it to the site of injury or infection. And it's very effective to allow immune cells to find their target. So the cells are going to swarm from an area of low concentration to an area of high concentration of chemotactic agents, and therefore get to where they need to do their jobs. So now, what are the benefits of the demon chemotaxis? You're going to allow the inflow of fluid that is going to dilute some of those toxic substances created by the microorganisms and the area of infection. You're going to be able to get fibrin to clog the area and trap the microbes and prevent them to spread to other anatomical areas of the body. Neutrophils and macrophages are going to be able to get out of the blood vessels by diapedesis and become to phagocytose and destroy the bacteria, be able to repair that tissue and pick up the particles that are all over the place, and then eventually the macrophages are able to fix and repair the area. So the pus that is often found after the area has been found by the fibrin clot, it's the mass of cells, liquefied cellular debris, and bacteria that is accumulated after the immune cells have encapsulated the area of infection around the fibrin clot. So there are certain reactions that tend to be long-lived in inflammation. The attraction of the monocytes, lymphocytes, and macrophages to the area. The macrophaging going around, clearing the pus, the cellular debris, the dead neutrophils, the dead red blood cells, and repairing the damaged tissue. The B cells coming in and being able to pick up some of the foreign molecules by the antibodies. Then getting stimulated to produce antibodies that can be used in the immune response. And the T lymphocytes going around seeing if a cell has been infected and being able then to kill those infected cells. And eventually the tissue is repaired and you have a scar that is left in the area of the inflammation. So now fever is one of those effects of inflammation, and is usually a good thing to have, especially in the low-grade area, which goes to 37.7 and 38.3 degrees Celsius, which is about 101. That a lot of microorganisms are mesophiles, and they do not like to have living in high temperatures. So the fever tends to do things to them that make them extremely uncomfortable and put them out of the temperature of their living. So it's a nearly universal symptom of infection, and is often associated because of the effects of the immune system with certain allergies, cancers, and other illnesses. The problem comes when we're having high fever. That fever that is 104 degrees Fahrenheit or above. That tends to be universally a bad omen when we're thinking about disease. But a fever of 102 and 103 moderate tends to be the limit that you will have before you can take a person to the hospital, to the emergency room. So now pyrogens, pyro from fire, gen for generation, are molecules that are able to generate fever. And some of those are coming from the outside of the body. One of the ones that we talk about are products of bacteria, viruses, or other microorganisms. In bacteria, the most important one is going to be the endotoxin, released from gram-negative bacteria when they die. But oftentimes, they can come from blood products that is being given to a patient, like in a blood transfusion, or plasma, or vaccination, or injectable solutions. Endogenous ones are the ones released by the immune cells, the monocytes, neutrophils, and macrophages during the process of phagocytosis and the induction of the inflammatory response. And of those, interleukin-1 and the tumor necrosis factor are the most important. Now, fever is beneficial because it attacks and impedes the bacteria from growing. Remember, bacteria want to be growing at a very narrow temperature, so increasing the temperature, it's going to cause problems to the microorganisms. Therefore, they're going to have problems with their nutrition, reducing the availability of iron. There's going to increase their metabolism, and it's also going to stimulate the immune response and other protective physiological processes to then help destroy the microorganisms. So fever has its benefits. The problem, as I mentioned to you a moment ago, is when the fever goes to medium and high levels. So oftentimes the experts say that if you have a slight to moderate fever, it's okay to let the person have the fever and let it run its course, because the potential benefits are going to be greater than the minimal side effects that you have. But when the fever goes into the high degree, now, especially when you have it prolonged, that's going to cause a risk that should be treated, and therefore that patient should be seen by a medical doctor immediately. So fevers that are associated with cardiovascular disease, trauma of the head, seizures, and respiratory illness should also be treated because of the nature of the illnesses that is causing it. But most people may think that having a slight to moderate fever is actually beneficial to fight infections. As I mentioned to you, another one of these molecules that we're going to be looking at are going to be the antimicrobial proteins. And of those, we're going to be looking at interferons. They are very small secreted proteins released by white blood cells and some tissue cells. And we have used them therapeutically to fight bacterial infections, to fight viral infections and cancer. Now, they're often a mechanism of defense released by the cell who's being infected. So in the picture that I have here, I have an infected cell that is being attacked by a virus. The viral nucleic acids, it's going to be assembling, take control of the cell, and the molecules of the virus are going to help trigger the presence of the interferon genes that is going to then release interferon. Now, the neighbor cell is the one who's going to benefit from the interferon. The cell already infected by the virus is basically long gone, but the neighbor cells are benefit from it because the interferons are bind to the receptors, which give signals to the new neighbor cell to synthesize antiviral proteins, and therefore it can attack the proteins of the virus and block replication of the virus, protecting the cell from the viral infection. So interferons have been shown to be extremely effective against viral infections and cancer. Now, they are general molecules not specific to a particular virus. So their synthesis is always in response to a type of infection, and it's always viral infection. So now we know their genes and we can produce this industrially to treat a lot of different types of virus infections and autoimmune diseases as well. Now the last antimicrobial proteins that I want to talk to you about, it's called complement. Complement is a protein system that is found in our plasma, composed of about 30 different blood proteins, and it initiates a cascade of events that eventually lead to the destruction of bacteria and viruses. So this cascade reaction is tightly regulated, so it doesn't happen without a trigger. And it is always happening after a physiological process initiated. So the first substance is usually a bacterial or viral molecule that is going to initiate the cascade, and then the first part of the cascade activates the next, then that one activates the next, and then that one activates the next, until at the end you form a pore called the membrane attack complex on the surface of the target cell that is infecting the host. So what we have over here in this slide are the three different ways in which complement can be activated. There's the classical component pathway that begins when an antibody molecule binds to a microbial cell via one of the PEMs. There is the lectin activated pathway that is activated by mannans, which are sugars. And the alternative pathway, it's one in which the cell wall components of fungi, bacteria, or viruses and parasites trigger an immune response on the host to initiate it. In any case, all of them are going to happen an initiation. An initiation, it's going to induce, in the classical pathway, for example, the activation of the complex C1 complex molecule, which, as you can appreciate in this picture, looks like a little octopus with many arms. Those are able to bind to antibodies. And when they bind to antibodies, they're going to initiate the cascade. That it will be the triggering mechanism. The cascade becomes active, and in here you have multiple proteins, some of them called C2A, C2B, C3A, C3B, C4, C5, C5A, and C5B. Anyway, this allows for the formation of a membrane component system that is initiated by C5 protein. So, not to go into a lot of detail, the molecules of C1 is going to activate the C4, C2, C3 complex, and that then is able to recruit C5 to initiate a membrane component. Once in the membrane, you have a polymerization event that is going to recruit other components of the complement system. Here you recruit C6, C7, C8, and the most important one, C9. C9 is going to make pores on the membrane, creating the membrane attack complex. And the membrane attacks complex, as you can see in this picture to the right, creates hundreds of holes on the membrane of the target cells, which now cannot prevent from leaking everything inside the cell to the outside. And therefore the cell then gets sliced and killed. So in this couple of tables, you have the triggers of the complement system. So again, the classical system, the lectin activated system, and the alternative system. You have the microbial activators that are going to initiate the cascade, and you have the molecules that are going to activate it. And then you have the cascade that eventually form the membrane attacks complex. Iron is an important molecule for respiration. So the cells who have iron are able to metabolize their energy via respiration and get a lot of energy via ATP. So therefore, we always tend to think of iron as a limiting factor that are going to prevent the growth of bacteria. So bacteria will generate iron binding proteins to try to sequester the iron from our cells. On the other hand, we have our own iron binding proteins to keep iron tightly bound and therefore not available for microbial use. On our side, we have hemoglobin, which, as you know, is the main molecule that is able to bind iron in red blood cells. But also, we have three other molecules worth mentioning: transferrin, lactoferrin, and ferritin. All of them are able to bind iron with a lot of affinity and sequester it away from microorganisms. On the other side, the bacteria have siderophores, which are also molecules able to often snatch iron out of the hands of our own iron binding proteins. So those are the ones that the bacteria uses to scavenge the iron away and take it from the iron binding proteins that are found in us. The last group over here, it's going to be the antimicrobial peptides, which are often secreted in sweat, for example. They are a large family of molecules, of which bacteriocins and defensins are some of the most important ones, and they are a non-specific innate immune molecule able to destroy bacteria by creating holes on the membranes of them. So as I see over here, we have, for example, a human antimicrobial peptide like a defensin, which is able to bind to the membrane of the bacterial cell and therefore create a little portion that is now going to allow the bacteria cell to burst. With that, I will leave you. This is the last part of the material for chapter 14 that I want you to cover, and I wish you a happy Thanksgiving, and I'll see you next week after the break. Bye-bye.